SwayamPrabha. Digital India. Educated India.
Welcome to the Proteomics course. In the previous lecture, we started discussing about the gel based proteomics. In that workflow, we talked about two-dimensional gel electrophoresis, and there are different steps which are involved in performing the two-dimensional electrophoresis. As I mentioned, the gel based proteomics includes various techniques such as SDS page, blue native page, two-dimensional electrophoresis, and advanced forms of electrophoresis such as difference in gel electrophoresis or DIGE. In the last lecture, I described you to perform a two-dimensional electrophoresis experiment, you need to follow a workflow, and I gave you different steps which are involved to perform the 2D. The first step was isoelectric focusing, which means you want to focus the proteins based on its isoelectric point. I have demonstrated you some video to give you the laboratory feel of the experiment, how it can be performed, and now we are continuing from the previous lecture. In today's lecture, we will talk about the workflow of two-dimensional electrophoresis. The first step was already described in the last lecture, and then we will discuss few more steps today. So, today's lecture we will focus on equilibration step, how to equilibrate the IPG strip, how to perform SDS page, which is protein separation in second dimension based on the molecular weight. Now, fourth step, the staining procedure, how to visualize your gel, because many time you have problems of low abundance protein, or sometime you are lucky and you have good protein amount. So, you can try different type of staining methods depending upon your requirement and your protein yield. We will discuss different type of staining methods which can be used to perform this step. So, let us start with the first point, equilibration. As briefly I described in the previous lecture, once you have performed the isoelectric focusing or IEF, you need to prepare your IPG strip for the next step, which is SDS page. When you are doing the conventional SDS page, you pour the gel, both resolving and stacking, and then you want to make some wells by using a comb, and then you load your protein samples in different wells. Now, when we talk about two-dimensional electrophoresis, it is different than only doing the SDS page alone. The concept of SDS page will be involved, which will be same, but here there will be variation. You do not need to add the staking gel. You need to prepare your protein sample, which is already focused inside the strip in the first dimension. Now, this IPG strip, which was already put in the electrophoretic unit in the IEF instrument, after that you want to prepare that so that protein can be separated further based on the molecular weight. Now, you need to add SDS to provide the negative charge, and you need to do some treatment for reduction and alkylation. When you are taking the protein sample alone, you have flexibility of boiling the protein sample in the tubes and do all those treatments to denature the protein separately. But now, your protein is already focused inside the IPG strip. So, you do not have that flexibility here. So, all you need to prepare your IPG strip in such a way that you can apply that on top of a SDS page gel. So, I will show you what steps are required in the equilibration step. So, after the first dimension separation is done, you need to coat the protein with SDS page. For making it compatible for SDS page, so you need to coat with SDS, sodium dodecyl sulphate, which is an anionic detergent, and it provides negative charge later on for separation in the electrophoretic field. The equilibration step performs cleavage of both inter and intra chains of disulfide bonds, and it alkylates the sulfhydryl groups of the cysteine residues. So, there are two equilibration steps which are required for this preparative step. The first equilibration step and the second equilibration step. Your recipe for equilibration buffer is ideally similar in both first and second equilibration, which includes 6 molar of urea, 2% of SDS, 0.375 molar of trace HCl, pH 8.8, 20% of glycerol, and then you need to make one variation in this buffer composition. Either you add DTT dithiothriotol or you add IAA iodoacetamide. Please note, this recipe is given only for your reference. People try different slight modification of this concentration and recipe for doing the equilibration step. Now, as I mentioned, in this buffer you can add DTT, and that will be used in first equilibration step. The first equilibration step can be performed for 10 or 15 minutes depending upon your strip length. Now, once you have done the first equilibration, you need to remove that solution and add a new buffer for second equilibration. Now, this buffer recipe is also exactly similar to the last one, except one change. DTT is replaced with iodoacetamide. So, you can add 135 millimolar of iodoacetamide during the second equilibration step. Now, this is very important because IAA can alkylate the residual DTT and it minimizes the vertical streaking. It also prevents the reoxidation. So, you want to really ensure that your protein is denatured again after doing the IEF process. It is coated with SDS molecule, so that now you can separate the proteins in the second dimension based on molecular weight. This step is also performed for 10 to 15 minutes depending upon the strip length, and once both the steps are done, then you are ready to perform the SDS page. I will show you in this slide the brief stepwise procedure how you can do the first and second equilibration. So, first strip can be equilibrated in the first equilibration buffer by adding inside a tray in a well, and then you change that with the second equilibration solution. The DTT reductant will be used for the first step, followed by IAA iodoacetamide, which functions as a scavenger of the excess reductant and alkylates and prevent the reoxidation. So, here you can see how different bonds are broken from the disulfide, and IAA is preventing the reoxidation, alkylating these residues. Okay, after having discussed the equilibration step, now we can talk about how to perform sodium dodecyl sulphate polyacrylamide gel electrophoresis or SDS page. As I mentioned, when you perform SDS page alone for separating your regular protein extract, then you need to add both stacking and resolving gel. Now, in this case, when you are using SDS page only in the second dimension of 2-dimensional electrophoresis, then you do not need to add the stacking gel. you have already focused the protein in the gel in the first dimension based on the IEF. Now, the second dimension separation, you have already prepared the strip to be loaded on the SDS page gel. So, rest of the step can be uniform or similar to what one can expect in regular SDS page. So, SDS page is one of the very widely used electrophoretic technique which separates protein based on their size or the molecular weight. So, this molecule SDS or sodium dodecyl sulfate, which is an anionic detergent, it provides negative charge. It binds at a ratio of 1.4 gram of SDS per gram of protein to provide almost similar charge to mass ratio. Therefore, your protein separation will be mainly based on charge in the electric field. As I mentioned, there are steps required to prepare your protein for denaturation. So, you need to boil your proteins in SDS and beta mercaptophenol and DTT to denature the protein and to remove or break any disulfide bonds. Now, since you cannot do this step boiling step in your 2-dimensional electrophoresis, where you have already focused the strip IPG strip in the first dimension. So, you need to do the equilibration step. Rest of the steps will be very uniform. So, let me describe you the role of the components which are being used in SDS page. Acrylamide, it provides the matrix or gelling for making the gels. Now, bisacrylamide, that is an cross linking agent. By adding both acrylamide and bisacrylamide in a fixed ratio, one can make this gel by adding the gelling agent and the cross linking agent. But that itself is not sufficient to polymerize the gel. You need to add few more reagents, which include APS or ammonium persulfate, which initiates the polymerization process. Timid, that is a free radical stabilizer, it also promotes the polymerization of the gel process. So, beta mercaptophenol is not used in making the gel, but for your protein treatment to break the disulfide bonds. So, I hope you are at least able to understand the role of each of these components which are being used in SDS page, either during the protein preparation of doing the SDS page or making the gel to separate the protein. Now, once this step is done, you need to boil your protein sample if you are performing a regular SDS page. The negatively charged SDS molecule will bind to the folded proteins. It will denature to the protein and provide the negative charge. Once this protein preparation is done, then you can load this protein on SDS page gel, and based on the molecular weight, the lower size proteins will migrate the furthest and the higher molecular weight protein will remain near the top surface of the gel. So, depending upon the protein size, you can separate the protein based on the lower and higher molecular weight in this SDS page gel. You can add the standards or known molecular weight proteins to determine your unknown proteins. Since we are talking here about 2-dimensional gel electrophoresis workflow, I think it is important to clarify that in 2DE, when you use SDS page, you do not need to make these wells by adding a comb, because you already have a flat IPG strip which has done the first dimension protein separation. So, in this case, you need to make a flat comb, you need to make a flat well, so that your IPG strip can be placed on top of the SDS page gel. Again, to remind you, here you are only making the resolving gel for better resolution and separation of the protein. The stacking part is not done for the 2D, which has already been done during the first process of IEF. So, SDS page is one of the very commonly used method for determining the molecular weight of a protein of your choice or unknown protein which you want to know how much molecular weight it is. So, if you run the standard protein markers along with your unknown protein, you can determine the molecular weight of the unknown proteins. I am showing you one SDS page image in which from the left to the right you can see the first lane is standard or known molecular weight. In this well you can see there are different at least 5 or 6 bands are visible. These are the known molecular weights, sometimes starting from 150 kilodalton to 10 kilodalton of molecular weight range. Then we have different wells, and in each well unknown protein for which you want to determine the molecular weight can be loaded, and after staining with Kumasi blue you can see a pure band here in this case. So, one can determine the molecular weight by performing the SDS page. I will now describe you the animation and show you the steps involved in SDS page. Sodium dodecyl sulfate SDS polyacrylamide gel electrophoresis SDS page. In this animation you will see various steps involved in separating proteins based on their molecular weight. First of all, the polyacrylamide gel containing SDS is cast between the glass plates as a vertical slab in the same buffer that is used for electrophoresis. The molecular dimensions of the pore can be controlled by varying the amount of NN methylene bisacrylamide with free radical cross linking being facilitated by ammonium persulfate or APS and timid. Next step is to create the wells on these gels. The sample wells of uniform size, shape and separation are made using a comb which is placed in the gel as soon as it has been poured. After the gel has polymerized, the comb is removed to provide the gel ready to start the process. Now, you can prepare the sample to load on SDS page gel. SDS is a negatively charged anionic detergent that binds to protein molecules and causes them to denature. The DTT used breaks any disulfide linkages that may be present. During the process, you boil or heat your protein samples to denature the protein. Now, the negatively charged SDS molecule will bind to the proteins and denature them. The binding of SDS causes the proteins to have uniform charge to mass ratio, thereby allowing the separation purely on the basis of molecular weight. The protein samples can be loaded on the wells by using a micropipette. As you can see in the animation, you can use different wells to load different type of samples. Once all the samples are loaded, then you can take this unit and move into an electrophoretic apparatus. A direct current supply between 100 to 350 volt is passed depending upon the size of gel for a time sufficient to separate the protein mixture into discrete bands based on their molecular weight. The progress of electrophoresis can be observed with the help of tracking dye. the larger proteins are retarded in the gel and remain close to the point of application, while the smaller proteins migrate further along the gel. The gel can be then stained with either Kumasi or silver stain and viewed to observe the various discrete protein bands present in each of the sample. For example, the left image here is silver stained gel and the right image is of Kumasi stained gel. You can load the molecular weight marker or the standards as shown in the first and the last and the 10th well, and then a known protein sample for which you want to determine the molecular weight can be separated in different wells. So, I hope after looking at this animation, now you are clear how to make a gel to separate the proteins based on their molecular weight, and I will show you later on one laboratory demonstration how to use SDS page gel for separating proteins in 2-dimensional electrophoresis. So, let us now move to a variation of SDS page known as blue native page, blue native polyacrylamide gel electrophoresis. Now, in SDS page we are providing a denaturing environment, we want to denature the protein, whereas in the blue native page we want to separate the proteins in their native condition, and by applying these two techniques together by using SDS page and blue native page, one can obtain very unique and different information often related to the isoforms, subunit composition, molecular weight and different type of post translational modification. This information can be obtained either alone from SDS page or from blue native page, or by combining these two together to get the integrated information. So, I will briefly describe you here the blue native page. Please keep in mind this is not going to be used for 2DE, but often people also use the native form for the 2DE separation. So, in the blue native page the protein analysis is performed under the native condition. Here you are not going to boil your protein sample with SDS and providing the different type of denaturing condition. So, you need to add a sample with Kumasi dye, which provides the necessary charge required for protein complexes to separate in the gel. But as I mentioned, unlike SDS, this dye will not denature the proteins, but it will bind in their native state itself. People apply the 2D blue native page for various applications, such as studying about the multi-protein complexes or MPCs. So, identification of multiple protein complexes is not possible by using the SDS page or denaturing environment. So, MPCs can be identified by using the blue native page. In blue native page, the electrophoretic mobility of MPCs or multi-protein complexes determined by the negative charge of bound Kumasi dye and size and the shape of complexes. So, this technique can provide the integrated view of protein function. I will now show you an animation how to perform the native polyacrylamide gel electrophoresis. Blue native page or BN page. The polyacrylamide gel is cast between the glass plates as a vertical slab in the same buffer that is used for electrophoresis. The gel is prepared by free radical induced polymerization of acrylamide and bisacrylamide in a suitable buffer. Ammonium persulfate and timid are added to facilitate the generation of free radicals and cross linking. The molecular dimensions of the pores can be controlled by varying the amount of an N-methylene bisacrylamide. Once the solution is poured, then sample wells can be created. The sample wells of uniform size, shape and separation are made using a comb which is placed in the gel as soon as it has been poured. After the gel has polymerized, the comb can be removed to provide a gel ready for the process. Sample preparation. The protein sample present in a suitable buffer is mixed with Kumasi blue dye, which provides the necessary charge to the protein complexes, thereby facilitating their separation in the gel. Unlike SDS, the dye does not denature the protein, but binds to them in their native state. The protein samples are then loaded into the wells with the help of a micropipette. As it is shown in the animation, you have different wells to load different protein samples. You can use one of the wells to add known or standard markers, and other wells can be used to separate the unknown proteins. Once all the samples are loaded, then this unit can be placed into electrophoretic unit and a direct current supply of around 100 to 350 volt can be passed depending upon the size of the gel for a time sufficient to separate the protein mixture into discrete bands based on their mass charge to mass ratio. The progress of electrophoresis can be observed with the help of tracking dye. The larger proteins are retarded in the gel and remain close to the point of application, while the smaller proteins migrate further along the gel. The gel can be then stained with Kumasi brilliant blue and viewed to observe various discrete protein bands. After watching this animation, I hope you are able to understand how to perform native polyacrylamide electrophoresis, and these concepts can be applied for using the 2DE as well. So, now you are familiar that how you can apply both SDS page and blue native page, and you can obtain unique information about subunit composition and different type of isoforms from different proteins, and you need to apply both the techniques together. Now, you are also familiar how to determine the molecular weight of an unknown protein. So, as I mentioned, you have seen the classical SDS page and blue native page. The concept is similar and it can be applied in the two-dimensional electrophoresis. There is little variation here, because now you do not need to add a comb. You have to have a flat comb for making a well to add your IPG strip, and you do not need to add the separation for stacking gel. So, in this laboratory demonstration video, I will show you for the two-dimensional electrophoresis how to make the SDS page. So, equilibration process will be demonstrated, as well as how to run the SDS page for 2DE.
Protein separation by SDS page. SDS page, which constitutes the second dimension of two-dimensional gel electrophoresis, involves assembly of the gel apparatus, gel casting, equilibration of the IPG strip, followed by placement of the IPG strip on the gel and protein separation. Assembly of gel apparatus and gel casting. Clean all the components of the gel assembly thoroughly with water and dry them with paper towels. Carefully arrange the glass plates in the gel casting assembly, interspersed with separator sheets depending on the number of gels to be run. The assembly should be tightly packed such that there are no leaks. Close the casting assembly and tighten the screws provided. Prepare the gel casting solution consisting of acrylamide bisacrylamide, tris chloride, SDS, APS and timid, and pour it with the help of a funnel into the central channel of the casting assembly. APS acts as a free radical generator while timid functions as a catalyst for the polymerization reaction. Ensure that the solution spreads evenly such that the top edge of the gel is uniform. Allow the solution to stand for polymerization to occur and spray a solution of SDS across the top to ensure that the gel does not become dry. Equilibration of IPG strips. Place the thawed IPG strip in the tray and over it pour the first equilibration solution consisting of urea, tris HCl of pH 8.8, glycerol, SDS, bromophenol blue along with dithiothiotol or DTT. Place the assembly on a mechanical shaker for 10 minutes. The dithiothiotol enables cleavage of all disulfide bonds present in the proteins on the IPG strip. Next, add the second equilibration solution to another well of the tray. This solution has the same composition as the first, except that the DTT is replaced by iodoacetamide. Transfer the IPG strip into this solution after draining off any excess liquid from the first. Again, place the assembly on the shaker for 10 minutes. Iodoacetamide prevents reformation of the disulfide bonds by binding to the free sulfhydryl groups that were obtained after DTT treatment. SDS page. Drain out the excess liquid of the second equilibration solution from the IPG strip using a tissue paper. Soak the strip for a few minutes in the same buffer solution that is to be used in the gel tank. Then carefully place the strip in between the glass plates and gently push it in until it rests firmly on the gel surface without any gaps. Place the filter paper containing the molecular weight marker beyond the positive end of the strip. Then pour the lukewarm agarose overlay solution over the strip to prevent the gel from drying up. Assemble the remaining plates in the gel assembly and carefully lower the entire apparatus into the gel tank containing the buffer solution. Ensure that the buffer level does not rise above the mark indicated. Place the separating chamber above this and pour the buffer solution into the upper chamber. Close the gel assembly and connect the apparatus to the electricity supply. Set the appropriate voltage and begin the run. All the proteins present on the IPG strip carry a large negative charge due to the action of SDS, which denatures the protein and binds to the polypeptide backbone at a constant weight ratio. This ensures that the proteins present get separated exclusively on the basis of their molecular weights rather than their mass to charge ratios as in a native page. The proteins having lower molecular weights have high mobility and migrate further through the gel pores, while the high molecular weight proteins remain closer to the point of application. This allows efficient separation of the proteins in the second dimension.
So now we will talk about how to stain the gel and visualize the protein spot, because so far now you are able to separate the protein based on their pi, based on their molecular weight, and now everything is on a transparent gel. You do not know how well your separation has occurred. So now you need to visualize your spots. So different type of staining methods have been developed, and those can be applied to visualize your protein bands or your protein spot, depending upon what type of gel you are using. So after giving you an overview of staining techniques, let me describe you few staining methods in more detail. Let us start with Kumasi blue staining. This is one of the most commonly used stain for protein detection in polyacrylamide gels. The sensitivity is good, but not very sensitive as compared to the silver or cypro ruby, but its ease of performing the staining method, stability, the cost very less, and good compatibility with mass spectrometry. All of this makes Kumasi blue as a stain of choice for most of the laboratories worldwide. Now, there are some advanced form of Kumasi stains being developed, including biosafe Kumasi stain, which is non-hazardous, made keeping in mind the environmental scenario. So, it can be disposed as non-hazardous waste. The biosafe Kumasi is also ready to use stain and a single reagent protein stain. So, I will show you one animation how to perform the Kumasi blue staining. In this animation, I will show you how the staining can be performed by using Kumasi brilliant blue stain. The completed electrophoresis gel is placed in a tray containing the Kumasi blue staining solution, typically R250, that has been dissolved in an aqueous solution of methanol and acetic acid. The negatively charged Kumasi dye interacts with protein through ionic and non-covalent interactions. After adding the stain, the tray is then placed on a mechanical rocker, which allows for uniform contact of the gel with the solution by means of gentle rocking. After overnight stain, you have to drain out the staining solution. The stained gel is then placed in a de-staining solution. Which consists of 50% methanol and 10% acetic acid to remove any excess dye that may be bound to the gel. Again, this process involves overnight strips or at least 10 to 12 hours of gentle shaking on the rocker. The stained gel can finally be viewed as shown in this image here, showing a 2D gel stained with Kumasi brilliant blue, and it can be scanned by using a scanner, and these protein spots can be easily viewed. So, in this animation you are able to see, after performing the 2D electrophoresis, how to add the Kumasi stain on your gel, do the staining and de-staining procedure, so that you can visualize your blue spots on the transparent background. Each of the spot can be now used further for image analysis purpose. So, most of the time, if you are lucky and if your protein extraction protocol is good, you have good protein yield, but often your samples are very challenging. Despite your good extraction protocol and your good laboratory skill, still you are unable to obtain a very good protein quantity. So, how to move forward? You want to still do the separation of the protein, but Kumasi stain may not be sufficient to visualize the spot on the gel. Now, you need few more sensitive stains, such as silver stain. So, I will describe you how to do silver staining. The silver staining is more sensitive than the Kumasi blue dye. First of all, when you have run your 2DE gel, you need to do the protein fixation in the gel by adding methanol and acetic acid. The staining procedure involves silver amine complex, which is bound to the tungstosilicic acid. The silver ions are transferred from the tungstosilicic acid to the proteins. Now, by providing alkaline solution with formaldehyde, it can reduce the silver ions to the metallic silver form, and the brownish tinge you can see on the spot, which can be further analyzed. But if you allow this reaction to occur continuously, you will see a very dark background on the silver stained gel. So, you need to stop the reaction by adding acetic acid. I will now show you this animation how to perform the silver staining procedure. In this animation of silver staining, I will describe you the procedure how to stain the gel by using silver stain. The completed gel is first placed in a fixing solution of methanol and acetic acid that fixes the protein bands in the gel and minimizes any diffusion. This step must be subjected to gentle shaking for around 30 minutes, after which the silver stain solution is added. After adding the silver solution, the gels are rocked gently in order to allow for proper and uniform staining. The silver stained gels are first washed to remove any excess stain and then placed in a developing solution where the silver ions get reduced to metallic silver. Formaldehyde in an alkaline solution in the presence of sodium carbonate or other alkaline buffers are commonly used for this process. Again, a gentle rocking will allow for uniform staining pattern. Finally, the gel can be viewed as dark bands or dark spots against a light background. As you can see in this 2D gel image, the spots having the dark background are stained with silver stain. So, let us see what we have learned today. In summary, we started with two-dimensional electrophoresis workflow. The first step, the equilibration we discussed today, isoelectric focusing was discussed earlier in the previous lecture. Then we talked about how to perform the separation in the second dimension. During that course, we talked about SDS page and blue native page. Then finally, we talked about different type of staining methods available. We looked at in more detail about Kumasi staining and silver staining. In the next lecture, we will continue with this workflow of performing two-dimensional electrophoresis. We will talk about few more sensitive stains available, how to use those, then how to analyze the images by using different software, and then to perform the statistical analysis of those, so that we can identify the interesting protein spots for a biological question. I hope now you are able to follow the workflow of 2D and will continue in the next class. Thank you.